Hey folks, this is the first part of a two-part series. We're going to go through mitosis in this video, and then we'll go through meiosis in the next video. So if you're here, you're starting at the right spot. So let's talk about mitosis. Mitosis, let's take a step back with it first and talk about how the general process of mitosis actually works. So a lot of people will like to start off with a circle with this diagram here, and they'll like to break it up into a couple different parts. So we'll break this up into a couple different parts here. So there's one part there, another part there. Draw this part like that. Draw this part here, and then we need one more line here. And so the way that a lot of people like to break up mitosis is into a bunch of different stages. And one of the main stages, or one of the ones that takes the longest amount of time, is this red inner piece here, which is all known as interphase. And interphase takes place in a series of different steps. The different names of those steps are G1, S, and G2. And basically what happens in G1 is this is sort of where everything starts, and so I like to call it like the rest stage. There's some cellular activity happening, but there's no real progress being made towards, hey, let's go ahead and try to make more cells. The overall goal of mitosis is you want to be able to generate identical daughter cells. You want to be able to take one cell and duplicate it exactly, sort of like copying and pasting on your computer, where you take one thing, exactly copy it, and paste it to a new location. So that's the end goal. G1 is sort of the rest stage. There's activities happening, but not real any mitosis-related progress. In the S stage, that is where we are going to duplicate something known as the chromosomes. And the chromosomes are just basically bundles of DNA that contain all of the genetic information for this cell to exist. Once we get past the S stage, we get to the G2 stage, and I often call this the Santa stage, because what happens here is in the G2 stage, the cell is going to check all of the chromosomes and say, hey, have I duplicated the information properly? Have I actually copied everything exactly as it needs to be? And it'll get rid of the cells or the genetic information that was wrongly copied, and it'll keep all of the good stuff and sort of put a check mark beside it and say, yes, this one's good to advance. So G1, S, and G2 are all known as interphase. And then after that, we actually enter the stage of mitosis. Following mitosis, we'll talk about this one later, but we actually talk about something called cytokinesis. And so we've talked a lot about these chromosomes. Let's draw what an actual chromosome looks like. So as I said, they're bundles of DNA. And so the way they're often represented in textbooks and in teachers' drawings is something that looks like this. And some teachers will put a circle in the middle here. And there's a couple of regions that we need to be concerned about with these. At the top here, we have the telomere. And the telomere doesn't really code for anything. It's mostly just like a protective cap, similar to like a pen. So my drawing tablet pen here has a cap to protect the little nubby at the end. The telomeres don't really have anything that codes for stuff super important. We're researching and discovering a lot more about those telomeres every day. But they are basically the protective cap. This middle part here is known as the centromere, and it's what's holding together, centromere. It's what's holding together these two long parts, which actually get broken in half this way. And these two long parts are known as the chromatids. And so throughout the process of interphase, we'll actually duplicate this chromatid, and so both sides of this should be the same and contain the exact same genetic information. So that's what a chromosome is. The stages of mitosis are broken down into a couple different parts. It starts with interphase, and then it moves into prophase, and then it moves into metaphase, anaphase, and the last stage is telophase. And then cytokinesis will happen after that. And so the way people remember this order is through the acronym PMAT. Now let's talk about what's happening in each and every one of these stages here. So the first stage, we kind of talked about interphase already. All that really happens throughout the course of interphase is that the genetic material is going to be duplicated. Those chromosomes are going to be duplicated. So in prophase, we have our cell, And the other thing that we have is we have our nucleus with its nuclear membrane intact. So we have the nuclear membrane or envelope, and it's going to be intact, or it's going to be there and it's going to have a function. And what we'll see is all of these duplicated chromosomes. I'm only going to represent and draw 3. See all of those duplicated chromosomes present in there. The other thing we'll start to see is something called spindle fibers or spindles. The other name for these is that they are centrosomes. We'll see those starting to form in the cytoplasm or in the space of the actual cell. These are the centrosomes. And we'll see those starting to appear. And what's going to happen over the course of prophase is that the membrane will start to dissolve. In other words, we won't any longer have that separation between the DNA, the genetic code, the genetic information, and the rest of the components of the cell. Those two are going to start to not be separated anymore. And so what will happen over the course of time is that in our cell, we'll see we advance to the next stage. And the next stage is metaphase. Metaphase is often better known as the middle phase, because what happens here is each and every one of those chromosomes that we had is going to start to line up in the middle. And those centrosomes that we talked about are actually going to collect at the side. And the way that I like to think about this is sort of like little spiders shooting out their webs. And these will be what's called microtubules. They'll shoot out their webs and they'll attach to those chromosomes at that middle part there, at the centromere is where they'll start to attach to those chromosomes. And so this is the metaphase, where everything lines up in the middle, and we have no nuclear membrane or envelope anymore. And so once that happens, we can then advance to the next stage, and the next stage is known as anaphase. Anaphase is just an extension of what was previously happening in metaphase, in that those chromosomes now are going to start to be ripped apart into their individual chromatids. And we still will have those spindle fibers sitting at the ends of the cell. They'll have shot out their webs, and they'll start to, sort of retract those webs and pull each of the chromatids closer to their own side. So we have this separation of chromosomes happening in anaphase. And so after anaphase takes place, we'll then advance to the next stage. I'm just going to give myself some more space here. We'll advance to the next stage, and the next stage will be for us to go into telophase. So telophase or telophase is the next stage that happens. And what happens here in telophase is that in our cell, which is now going to be really, really stretched out, it's going to be elongated like this. What's going to happen is at those ends, and maybe I should draw it a little bit more accurately. Let's draw it like stretched this way, since that's how we've been talking about it. At those ends, we're going to start to see the nuclear membranes starting to be reformed. So we're going to start to see those nuclear membranes starting to come back, and we'll have each of those chromatids now being pulled apart. So those are nuclear envelopes starting to be reformed, and we have each of those chromatids that were previously pulled apart. And that's what's starting to happen in telophase. And then as we advance one more time to the last step, which is cytokinesis. What we see is we see that cell actually starting to separate into two smaller individual cells. So we'll see that nuclear membrane present. We'll see all of those different chromatids now present inside those nuclear membranes. Again, those are the exact same chromosomes in both different cells. So now we have the exact same genetic information there. And what'll start to happen is this cleavage furrow is what it's called. Cleavage furrow will start to form. And these two cells will eventually start to separate. And the process of cytokinesis is that these two cells will separate into their own individual cells. And so the end goal is we'll have two exactly identical cells that we created simply by duplicating the genetic material, going through a series of steps, and now having these two identical cells. And that is how mitosis works.